So yesterday we talked about mutations and why they happen and what can be the result of them. Today we're going to talk about other changes that can happen to your DNA aside from mutations. We're going to start with prokaryotic cells. And as a general rule, prokaryotic cells are not the focus of our study. We're mostly talking about eukaryotic cells, which are the types of cells that you have. But we can't forget that that is not the only type of cell that exists. And so I'm starting this lesson with a few things that happen in prokaryotic cells to give variation in their DNA code. So you might remember me showing you previously that in prokaryotic cells, like bacteria, their DNA is circular instead of in linear chromosomes. So that's what's pictured here. And there are four things generally that happen to prokaryotic cells that could achieve variation. Some of them you have heard about indirectly or directly before. The first one is called transformation. And I could sum this up as the process of taking DNA. So on the picture that I have here, you have in the middle a bacteria. So this is your prokaryotic cell in the middle, the big oval-shaped thing. And on the transformation section, we can see a gene moving in to the bacterium so that it can incorporate and be part of its DNA. So transformation is when a prokaryotic cell takes some DNA from somewhere else. Transduction usually has to do with viral DNA. So on the bottom, there's a picture here of transduction, and you might recognize that little image. That is a bacteriophage. It's a virus that affects bacteria. I showed you one previously. This is when viral DNA is incorporated. And I'm just going to put a little note here about two people. Hershey and Chase were two people, two scientists, who were trying to figure out, is DNA the genetic material or are proteins the genetic material? And they used these viruses, bacteriophages, to insert DNA into bacteria cells to determine whether or not radioactive markers showed up. In one trial, they put the marker on proteins. In another trial, they put the marker on DNA. And they found that when they put the marker on the DNA, the bacterial cells also had this radioactive marker. That was an important experiment. That is an experiment that I would ask you about if I were a person who makes tests for Biology 30 that you are going to do tomorrow. The third thing on the list is conjugation. This is transfer between two cells. So it's illustrated over here. We saw this already in chapter 16. Bacterial conjugation was a prokaryotic's version of sexual reproduction, where two bacterial cells exchange some of their DNA. And transposition has to do with jumping genes. And you might remember seeing this word before as well. Transposons are genes that move around, jumping genes. So these are four ways, aside from a mutation, that a prokaryotic cell could have some variation in its DNA. Now, does anyone want to ask anything about prokaryotic cells or any of those four things there? So we're going to move on and talk about something that has to do with eukaryotic cells, although it's not exclusive to them. And we are going to talk about recombinant DNA. Now, we know already from our last chapter that sexual reproduction produces variation. That's fantastic. We're going to focus on some other ways that we can get genetic variation besides sexual reproduction. So the first thing we'll mention is genetic engineering and what that is. Genetic engineering is the manipulation of DNA, very simply. And usually the goal of genetic engineering is to modify genetic expression. So when someone is doing genetic engineering, they are changing or manipulating DNA in the hopes that when that DNA gets translated to messenger RNA, that messenger, or transcribed, that messenger RNA gets translated into a protein, I get a different result. I get different expression. Genetic engineering produces recombinant DNA. Now, recombinant DNA would be generally a new gene is inserted into a genome. So somewhere in your genome, which is the entire collection of DNA that you have, we would have inserted a new gene. Now this recombinant DNA could be in a eukaryotic or a prokaryotic cell. So this process could apply to both types of cells. And sometimes it's between the two types of cells. So how do we get a new gene inserted into a genome? We have to start with a restriction enzyme. And I'm highlighting the word enzyme because there are a lot of enzymes in this chapter, and something that would frequently be asked is, can you differentiate between all of the enzymes? Do you know what each of them does? There is more than one type. So restriction enzymes has an S on it plural because there are different restriction enzymes that do different things. All restriction enzymes catalyze the cutting of DNA at a specific, and I'm putting that in capitals because it's important, at a specific nucleotide sequence. So restriction enzymes are going to help cut DNA, but they're not just going to cut it anywhere. They're going to look for a very specific sequence and only cut there. So on my little picture, these little guys that look like kind of evil Pac-Mans, those are the restriction enzymes. In this example, the restriction enzyme is looking for this very specific code. GAATC on the top, that would be the coding strand, and on the non-coding or antisense strand, CTTAAC. That is the exact nucleotide sequence that this enzyme is looking for. It is not looking for something close to that or something similar to that. It is looking for exactly that. So this restriction enzyme will cut the way that I've shown. And this is what restriction enzymes do. They cut the DNA sort of unevenly so that they're cutting more off of one strand and maybe less off of the other strand. Now on the left, I have this sort of white-colored DNA. Let's call this our human DNA. And on the right, I have this black-colored DNA. Let's call that bacterial DNA. The restriction enzyme will be used to cut two different samples of DNA, but exactly the same way. So it will look for the same sequence on a sample of human DNA and on a sample of bacterial DNA. The result of a restriction enzyme are these two things down here. We get fragments, and we get fragments that have what we call sticky ends. Now the word sticky is being used not in a literal sense. They're not actually like glue, but the fragments that we make are going to have unpaired nucleotides. So these sticky ends are these little segments here. That is a sticky end. It's a section where you have a bunch of unpaired nucleotides. Now on the sample of human DNA, I will have a sticky end. On the sample of bacterial DNA, I will have the complementary sticky end. So these restriction enzymes will cut two samples of DNA the same way, so that if I were to put them together, they would complement each other. And that's where we get the word sticky from. We're saying that these two ends will stick together. They complement each other. If then we take those two samples of DNA that we have cut with restriction enzymes and we put them together, we could use ligase, so the enzyme that sticks nucleotides together. We saw it before when we saw DNA replication. And now we have recombinant DNA. Now in this particular example, what probably happened is that we put a human gene into the bacterial genome. Doing things the other way around would pose a lot of ethical concerns. So putting DNA into a human genome is a little bit trickier to do. But taking a human gene and putting it into the bacterial genome, that is something that happens. A really good example of this is the insulin gene. We can take a sample of human DNA, add restriction enzymes to cut out the insulin gene, and then put that into the bacterial DNA. The result is that now the bacteria will produce insulin, which is great because we need insulin for all of those people who require it because they are diabetic, and this helps maintain their blood sugar. So this is an example of genetic engineering. There are two enzymes, restriction enzymes and ligase to attach things together, that would be at play. You need the restriction enzyme to cut a gene out, and you need ligase to put the gene into or reattach it to your new genome. Does anyone have any questions they'd like to ask about that? Now this is a very generic overview. There are obviously lots of specific enzymes that do specific things. You need to understand the basic idea of the process. Now once I've cut my DNA into fragments, I could recombine it, or I could do something else with it, and that's what I'd like to show you on the next page. Sometimes, instead of modifying a genome, we are trying to analyze a segment of DNA. DNA analysis that we'll discuss is done with fragments. That means I used restriction enzymes. So those same types of enzymes that could cut out specific sequences so I could insert a gene into a genome can also be used to help me analyze DNA. So the technique that I'll show you is called gel electrophoresis. It's called gel because this sort of square rectangular box is full of gel, smushy, jello-y type gel. It's called electrophoresis because there will be a current. There will be an electric current running through this gel. Now, if any of you watch crime-type TV shows, you might have seen someone do something similar to this before. In gel electrophoresis, here's what happens. You take a sample of DNA, you get it into a little micropipette, which is just a little sort of measuring syringe-like device, and you insert that, or you put the sample in a well. So at the top of the gel, there are like little cutout spaces that look like a well, so it's a little empty space that's been carved out, and that's where you put the sample. The idea with gel electrophoresis is not usually just to test one sample, but to compare multiple samples of DNA. So the next thing you would do is add samples to compare. So over here, you can see we would be adding some more samples. Now, 3 is just how many is pictured here. That is not how many you have to use. That's just what the picture is showing. You could do 10, you could do 5. It just depends how big your gel electrophoresis apparatus is. Now, once you have all of your samples in the wells, then you apply a current. So there needs to be electricity, an electrical current, running through this gel. If you take a look, you can see negative on the top, positive on the bottom. That's kind of a small detail. When you apply the current, fragments of DNA move based on size. And the idea would be large fragments move slowly and small fragments move quickly. So as the electrical current is running through the gel, the little fragments that were in my sample of DNA, and they were there because I used restriction enzymes, will start moving from the top towards the bottom of the gel. Now in the long run, my small fragments will generally move very far, so they will move close to the bottom of the gel, and my large fragments will stay closer to the top of the gel. Now the result that you get is what we call a DNA fingerprint. And this DNA fingerprint can be used to identify you or whoever's DNA we were testing. Perhaps it will identify you as the criminal who left some of their saliva at the crime scene, or perhaps it will identify you as the son or daughter of the person whose DNA we were comparing you to. So it can be used to identify. Common uses of this would often be in criminal cases or family-related things, like are you related to this person, yes or no. Fun story here. I read an article once about this lady who, I think she was getting a divorce from her husband, whatever. She wanted child support from this man. And so they were going to do some DNA testing to prove that he was in fact the father of the children so that she had a legitimate claim to collect this child support. So they do the test. Of course, the man is the father, but the tests show that she is not the mother. And she's like, that's ridiculous. I gave birth to these babies. How can I not be their biological mother? And it so happened that she was pregnant at the time. And so they did some sort of legal thing. I'm not sure what the lawyer did. I don't think he watched her give birth to the baby. I think there was some sort of affidavit that got signed. But she had this third baby. They tested this baby that she had very obviously given birth to, and that baby also was not her biological child. And I think I might have mentioned this briefly at the end of a class once. It turns out her ovaries were actually her parasitic dizygotic twin that didn't survive in utero, but that she was actually having her sister's babies. Even though this twin was never born, she didn't actually have a sister. But because in utero there were twins and one of them got absorbed by the other, she actually had two different people's DNA, her own DNA in her body and different DNA in her ovaries. Super crazy. Does anyone have any questions that they would like to ask about this so far? I heard about that and I wondered, maybe that's why I don't look like anyone in my family. Maybe my mom actually has her parasitic twins ovaries and that's why I look different. But that's a story for another day. If we look then at this picture of a gel electrophoresis that has been run, I will just point out, for example, this sample and this sample are the same. They have the same bands in the same places. So I can say that those two samples must have come from the same person, or perhaps from two people who were identical twins. I could say the same thing about this sample and this one. So generally, when you're looking at a gel electrophoresis, you're doing a very similar thing as to when you're trying to match up chromosomes. You're looking for how many bands there are, how big they are, how they're spaced out. Now, the DNA fingerprint doesn't just need to be for identical samples. Especially in the case of families, we are looking for similar results. If I were to run your DNA and one of your parents, half of your DNA should be the same. It might actually be more, but half of it should match because you got half of your DNA from your mother and the other half from your father. So I'm just going to pause here for a minute, discuss, and then we're done. The next thing that I'd like to mention is another technology. PCR stands for polymerase chain reaction. Now, DNA can end up in fragments. And when there is a fragment of DNA that perhaps we would like to make copies of, or perhaps we would like to study more, we can use polymerase chain reaction. It is a method to produce a large quantity from a small sample. So polymerase chain reaction is a method where you can take a small sample of DNA that perhaps has a sequence that you want lots of, like it's the gene for insulin and you want lots of copies of it, or perhaps you have a sample of DNA from a crime scene and you need to do lots of tests on it and you want to have lots of copies so you can do lots of tests. Maybe that's another time that you want to use polymerase chain reaction. You might even want to use it if you're studying a virus and you're trying to figure out how the virus works. You might have a tiny sample of it, but if you make lots of copies, then you can study it more completely. It's based on two principles, really. One principle is that the enzymes, all of the enzymes that we've been discussing, polymerases, ligases, etc., the ones that make copies of DNA, the ones that help with transcription and translation, the enzymes depend on temperature. So that's one thing. The second thing is that hybridization, which means the idea of complementary strands, is also temperature dependent. So basically we're saying everything about DNA making copies of itself and replicating itself all depends on temperature. So to use polymerase chain reaction, you can control the whole process by changing the temperature. So basically we're saying we can make some enzymes be active and some enzymes not be active, or vice versa, depending on what temperature we have the sample at. Now, does anyone have any questions that they would like to ask about technology, restriction enzymes, or what we can do with the fragments of DNA that we get from them? Gel electrophoresis or polymerase chain reaction? So that's a little bit of technology. There's a ton of technology in this domain that people would be welcome to use as their article presentation if they wanted to, for example. The last slide that I have to show you isn't really notes, it's a bunch of questions. One of the things that we need to do as we talk about biology is we need to think about society, and we need to think about perspectives. Now, a lot of the stuff that we talk about has some basic science, and the basic science, of course, will be part of test questions. But beyond that, there is a lot of discussion about should we be doing this, and what are some reasons that we should, and what are some reasons that we shouldn't. So the first question I put up here is the word property. A lot of the studies of DNA have sort of at the core, who owns the technology? If you discover something about the DNA of human beings, do you own that? Or because we are human beings, should everybody have access to that? So this idea of property brings up a lot of maybe economic questions, a lot of maybe even political questions, maybe even a lot of moral questions. Another thing that often comes into the discussion when we're talking about genetics is the idea of making clones. There are a lot of religious or moral or ethical concerns about making clones, but there are also a lot of maybe, say, health and economic perspectives that would counter that. So making a clone might not seem like a good idea unless you consider that making the clone could help us figure out how to cure a disease, or that making a clone could save us money because the clone is another copy of a plant that is producing food. There's the idea of transgenic organisms. So you might have seen GMOs, genetically modified organisms, organisms that have had genes inserted into their genome from a different species, like I showed you with the insulin gene. Obviously, there are a lot of economic benefits to this. Some things that have been done, for example, is there's a type of wheat that has been synthesized that is resistant to a herbicide. So you could spray the field with something that will kill weeds, but not kill the wheat that you are trying to grow. That's very economically advantageous for the people who are growing the wheat. But there might also be, on the flip side, some health concerns. What if you are allergic to shellfish, and then we take a gene from a crab and put it in wheat? Maybe are you allergic to wheat now? That would be a legitimate, real concern that people could have. Prenatal diagnostics is another thing that has to do with genetics. This would be the idea of screening for chromosomal abnormalities. So there are tests that can be done before a baby is born. to determine if they have Down syndrome, for example. And you might think, oh, well, that's a good idea, because you would want to be prepared. If your baby was going to be born with some sort of chromosomal abnormality, you might want to be prepared. And you think, well, that's a great idea. Who wouldn't want to do that? But on the flip side, you might have some real moral questions. Would this increase the rate of people wanting to abort a baby because they know that it's going to be born with this chromosomal abnormality? Then there's the idea of genetic therapy. So the idea of modifying your genome. So we're talking about people here. And anytime we're talking about people, there are a ton of sort of ethical concerns that come up. It is really difficult to do experiments with people or on people. There's not a lot of types of experimentation that are allowed. When I was in university, I did research and we used animals, and the amount of paperwork that I had to go through to test something on a rat was incredible. And so if you can imagine how people would value the life of a human versus maybe how people would value the life of a rat. And I'm not saying that one is better than the other, but it's incredibly difficult to figure out how things work in humans. And so there are a lot of people who have really strong opinions, either one way or the other, about whether or not all of this technology that goes along with genetics is a good thing to do.